during the day already. So I would like to apologize in the first place because you see, well, this is our environment. It looks very nice. That's actually where we are working in the center of London. But as always, there's a little bit of a construction site around me. And as a consequence of that, apologies for any noise that comes on top of my voice, yeah, throughout the talk. It's like there's some drilling going on. So it's like at the dentist, I'm sorry. So, well, the topic of the talk is tracing the magic bullet. The magic bullet means, well, it has been branded, engineered cell therapies have been branded in recent years as the magic bullet. This is a completely new dimension of a therapeutic. It's a life subject, if you like, if you are fascinated like me by cells. And it's something that is administered that can change. It's different to the previous drugs that we have been administering in the clinics. And that means, on the other hand, with a word of caution, that also we need to be careful about certain safety aspects that are related to administering these cells and what they may do that we may not have envisaged. So these are the words of caution. So where are we in the field? Live cell-based therapies are plentiful approved by now. So you see here on the cartoon at the end of 2022, that's data from the ASGCT, 60 live cell therapies were approved which are not genetically engineered, and 24 were already approved which are fall in the category of either genetically engineered live cell therapies or gene therapies. So these are, for example, your CAR T cells. But when you look into what is being developed, then the picture changes dramatically. You see that more than half of the products that are being currently developed are genetically engineered. Yeah, again, cell therapies and gene therapies, and we're speaking here about almost 4,000 clinical products or trials. And that means there is a massive boost in engineering these cells now. And you can imagine that questions are raised, of course, when you have an adoptive cell therapy product, and questions such as where does it go in the body? So like biodistribution, does it hit the target? Does it possibly relocate after administration? And where and when will off-target effects happen? And then there are questions, obviously, around whether these rather expensive therapeutics survive and how long they survive, and whether this has an impact, of course, on the treatment of the disease. So a lot of those questions that are here on the slide could, in theory, be answered by imaging methodologies. And, well, how are they answered now? How did the people get the product into the clinics now? Well, a lot has been done with biopsies. And a real advantage of biopsies is the fact that you can look at very many parameters. But biopsies come with some sort of drawbacks, such as that they are invasive, that they are expensive, complications are possible. In the context of cancer, for example, it has been shown that biopsying certain tumors results in enhanced metastasis. There's massively undersampling going on. So you take small samples, you get sample heterogeneity, and then, of course, repeat biopsying is a real challenge. So this is where the opportunities open up for imaging, whereby the drawback, obviously, of molecular imaging per se, and we're talking here about things like molecular MRI, but much more like spectrum, molecular PET, immuno-PET imaging, you can really only look at a few parameters, in most cases only one. But the advantage is you can do this repeatedly and quantitatively. It's reliable, it's diagnostic, you get whole body images out. And so I'm not pitching here imaging against biopsy, I'm pitching here imaging as to complement biopsies. So the vision is that maybe biopsying is going to be the tool that actually defines at an early stage of diagnosis what type of treatment you get, and then imaging, for example, could be the monitoring tool, the tool to predict that the therapy that you are currently getting is actually the right therapy, or whether you have to change things. So, thinking along those lines. So the question is, what are the available imaging modalities? Well, you are an imaging community. I don't need to say much to this here. That's a plethora of imaging modalities, and I just tend to put this up in order to show people what is out there, but also to make them aware that there is a set of imaging modalities like the one here on top that are actually used in clinical practice. And total body PET, as mentioned by my predecessor here, to be available in Finland, is available also now across several of European countries, including also in the United Kingdom, with 2 hubs, one in Edinburgh, one in London, at our college. And then you have technologies that are exclusively available preclinically. And that is, of course, something that needs to be considered when we think about tracing these magic bullets and these cell therapies, because we need to make the jump between what we can do preclinically for validation and what is potentially translatable to the clinic. So how would one actually approach the topic of tracking the cell therapy? Well, option 1 is a very obvious option. You may have an endogenous target on your adoptive cell therapy, and you try to image this endogenous target in a, for example, immuno-PET approach. So that, what does that mean? You basically generate a probe against the target. If that probe is a radioactive probe and you image it by PET because of the radioisotope is conducive with PET imaging, because it's, for example, a positron emitter, that would basically mean you have a classical immuno-PET approach. So is this possible? Has this been done? Yes. That is something that has been tried. So, for example, built on technology invented by Anna Wu's lab at City of Hope, previously UCLA. So they came up with a really nice cis mini body, which you can use to image CD8 T cells in humans by immuno-PET. And you see here an example courtesy of Imaginab, who is the company who has developed this companion diagnostic. And you see how nicely you can image the CD8 T cells in normal immune organs in this patient. And you see it in the tumor, which in this case of the patient is sitting down here. It's a melanoma patient. You see clearance of the tracer through hepatobiliary and renal routes. And you see very low background in other areas of the body. Of course, when it comes then to detecting cancers in areas that are not so low in background, for example, because you would have bone metastasis, then you can imagine, because of lots of other immune cells that you have in the body, it becomes a little bit difficult and challenging to identify your administered cells. So there are questions about adoptive cell therapy and whether it's specific. So in a late stage cancer patient who has received a lot of lymphodepletion, that is maybe not a big deal. But the situation looks very much different if we are talking about using adoptive cell therapy for other indications than late stage cancer patients, for example, like autoimmune disease, which is one of the hottest areas for cell therapies currently, and loads of investment and loads of new cars and applications in that space. So one needs to think about specificity really. And some people, some colleagues here at MGH, they have tried to do this. So they have said, well, let's image the targeting agent. So they have developed an antigen against the CD19 targeting agent. Yeah, so this is the single chain FV that is sitting as part of the CAR on a CAR T cell. And then they developed an CD19 ectodomain, essentially, which they put via some fancy chemistry onto a PET isotope and used it to image. And when you look at this preclinical data, which came out just like 3 weeks ago, you see this is quite impressive. A lot of specific uptake of the signal goes here in the spleen. This is exactly where you expect these cells to be in these models. So these are tumor models. And with Raji tumors and CD9 targets, lots of the bone marrow is exactly where you want to see these cells, so they are going there. You see in the control animals none of those signals. And then in order to interpret those images, please always keep in mind, of course, that radio tracer in nuclear medicine applications has to be cleared. So obviously you see kidneys and bladder and a little bit of liver here because of the way it gets excreted. But the probe is pretty specific. It's a real advance. And so what have they tried to do with this? Well, they showed us really nice sets of data where they administered different types of CARs to different tumor models. And they claim that they can actually predict how successful the therapeutic is depending on how much the CAR is expanding, for example, in the spleen, CAR-Ts. That's quite nice. It's obviously really nice to see this from a cell therapy developer perspective, because you can say, well, I have some tool now that tells me that my CAR T cells are expanding, are engrafting it properly, and that is obviously predictive for therapy success. And this is what they showed in this paper. So that is better, more specific than the CD8 probe. So it images, or is set to image exclusively the CAR T cells in this setting. Of course, it's not free of concerns. One of the concerns is, of course, what is the impact of that antigen probe onto the CAR T cells? Is it possible to sequester the CAR T cells from their intended target? Is it possible to activate the CAR T cells at off sites? Yeah, just because the antigenic probe is not targeted, it will go just to the CAR T cells, no matter where the CAR T cells are. What does it mean when you suddenly have a lot of CD19 active domains swimming around in an organism? What are the consequences on the immune system? So there are lots of questions to be asked, and it's really early stages, but it was a step in the right direction, I think. We will see where they take it at MGH from there. So what else can we do? Is this our watershed moment? Well, the alternative would, of course, building something into these CAR T cells so that we can track them, rather than try to track them after administration with some probe that is exogenous on the basis of whatever they express. Yeah. The question is then, well, and this is actually an image from Waterloo Station, so it's the biggest train station, a computer train station in London. And you can imagine it's difficult to find your friend amongst all of those people. So, and the way of finding your friend among all of those people is to generate contrast in those cells, not necessarily by exogenous administration, as I showed you before, but by some other way. So that is obviously the concept that lies behind all these efforts that people have done in order to generate bioluminescence traceable cells in the past. So that is great. But it raises the question, of course, well, what do we do with bioluminescence, for example, because you can't translate that approach. So I have here a little table for you, which lists the properties of imaging modalities, and they're sorted here by sensitivity, from the most sensitive ones down to the less sensitive ones, or the one that cannot be classified in this way, in an easy way. So, and the bioluminescence imaging has another huge advantage. This is really the most sensitive of those imaging technologies, as long as you can image it, because you can't image very deep, or you get all sorts of artifacts because of it's operating on the basis of light. There's scatter, there's absorption, so it's obviously a little bit of a problematic situation. What is the next best tool that we can use for the purpose of tracking our cells post-administration? Well, these are the radionuclide imaging modalities. Simply because you only need picomolar tracer concentrations, and you get the advantage that you can image at depth, so basically unlimited. You have somewhat better spatial resolution than bioluminescence, of course, too. Nowadays, you can multiplex even in PET. Currently, it's 2 isotopes. There's people who are trying to expand this. You can certainly multiplex at SPECT. The downside is maybe it's a rather expensive approach. But that's still the most promising for when clinical translation is actually an important parameter. Others have tried with MRI. The downside of the MRI is you need rather high amounts of contrast agent, and that is problematic. And I won't go into much detail on any detail today on this. But you should keep in mind an incentive to not completely rule out MRI is that it actually comes with superior resolution than the radionuclide imaging technologies. However, there is a sensitivity issue. So what is the methodology? Well, the oldest and simplest way to track cells is simply by radiolabeling those cells, administering the cells, and then observing the patient, the subject, the animal, and see what happens, where the cells go if they redistribute. So there's loads of label types available. So you have all sorts of radioisotopes, long and short half-life that you could put in. You just need to be careful with the dosimetry. You will see in a few minutes why. And of course, there are a few caveats. First of all, is label efflux. So your cells might want to get rid of the label. Then the second one, that's a big bummer, that is label dilution. Because when you have a fast expanding cell line, like this is actually what we want with a CAR T cell. We want to get it to the target. We want to have it expand at the target. And that means we'll lose the label because after a few cycles of expansion and cell division, obviously the concentration of the label halves every time. And as a consequence of that, you reach the sensitivity threshold of the detection technology and you can't see the cells that you're interested in. So the most interesting cells, which are those that are heavily expanding, are the ones that you lose first. That's a problem, in a conceptual problem here. And then, of course, there is no viability information. What has been tried with those approaches in the past was looking at the visualization of the delivery of a therapeutic. Here, this was 2 decades now ago, was I think done in Nijmegen. They could show whether they can, so that they can use SBIO labeled dendritic cells. They could look at the delivery. These are data from King's, where colleagues have looked at specific populations of eosinophils and neutrophils, and these 2 movies that you see here, they are actually taken on the same time scale. So you see how in the same patient at different times, eosinophils and neutrophils differently distribute within a given time window. So, and it's quite impressive. And the technologies around are not overly novel, yeah? So using radioisotopes for SPECT tracking or gamma camera tracking, as it originally was, is technology that is 40 years plus old. Yeah, and traditionally it was done with indium oxine, and nowadays it's largely done with technetium HMPAO, at least in the United Kingdom. It has decades of use for white blood cell scans to identify occult inflammation, and this is something that is just working. But because of the better sensitivity of PET instrumentation, people have been hunting approaches to do this by PET, but they were hampered initially with, because the standard isotopes that were envisaged weren't pretty good. So F18 has too short a half-life, copper-64 has significant label efflux from cells due to copper homeostasis in cells. And only when zirconium came into the game, tracking was enabled for about roughly 1 to 2 weeks. So the first people who have actually made that happen in the form of zirconium oxine tracking was Noriko Sato's lab at the NIH, who showed this. It's almost a decade in the past now. And then several other groups, including ourselves, have shown this in therapeutic immune cells. And like CAR T cells that you see, sort of City of Hope, and then gamma delta T cells, as we showed at King's. And then again, Noriko Sato with NK cells, in fact, in monkeys at NIH. And Susan Lapierre from Birmingham in Alabama has actually shown the first white blood cell scans now almost 2 years ago in the US. So adoptive cell therapy is still not being tracked, but we may see some distribution images in the near future. So this is where that field has gone. So I show here you a few examples of what we have been doing with these gamma delta T cells and what is the rationale of doing this. And so some of you may know that gamma delta T cells, particularly the gamma 9 delta 2 ones, they can quite nicely be expanded ex vivo, and they're envisaged as cell therapy because they can target so-called phosphoantigens. So if your tumor expresses phosphoantigens, then you get preferential targeting and activation of those cells, and they can actually kill tumor cells. The question is about the phosphoantigens. Are they there? What do you need to do in order to get them there? So, and there are so-called mevalonate pathway inhibitors that result in the upregulation of, for example, isopentenyl pyrophosphate, and as a consequence, targeting of those gamma delta T cells to the tumors. So we asked the question, do the gamma delta T cells reach the tumors? And after a few studies that we have conducted in order to optimize, again, imaging-based, so can read this up in this paper down here from Edmonds in SCS, we could optimize that the ideal scenario for liposomal mevalonate was to actually wait after administration for about 72 hours and then administer the zirconium-labeled gamma delta T cells. And afterwards, we followed up those animals. And of course, this needs a lot of quality control in terms of the cell labeling. You can't put endless amounts of radioactivity into these cells because you damage the cells. So you see that quite nicely here. It's important, really important, to look at viability not just after labeling, but look at how these cells proliferate afterwards, because otherwise it's difficult to detect any damage that has been caused. They remain functional, and we also did quite a bit of work in looking into the actual DNA damage caused. So you see here in green dots, you see basically instances of DNA damage, double-strand breaks. There's always a basal level of double-strand breaks in cells. That's normal, but at some point it kicks off and becomes significant. Then we put this into the bracket of somewhere between 20 and 50 millibecquerel per cell. Labeling with zirconium results in damage that cannot be rectified anymore. Yeah? So this is important to keep in mind. So we then radiolabel these cells at those levels. So in this case, it's about 20 millibac per cell, and administered those cells into animal models that also have had traceable, radionuclide traceable tumors. So that is a complicated image here. So you see two scales. So look at the one largely in green. You see the tumors here. This is because they are sodium iodide syn-porter traceable tumors. You also see a few endogenous sodium iodide syn-porter signals which you can ignore, like the stomach or the thyroid and salivary gland. Yeah? Let's focus on the tumor. And then you see on the purple to red-orange scale, you see zirconium signals overlaid. And all of that is overlaid onto CT. So the zirconium images, they tell you about the labeled T cells, gamma deltas in this case. So an hour after administration, unsurprisingly, you have basically almost all of them in the lung, a few start to be in the liver. 2 days later, all of them are in the liver, large amounts are in the spleen. And a week later, still in the spleen and a lot in the liver. When we zoom in onto the tumors, you can actually quite nicely see how these T cells go into the tumors. What you also see is that there is some heterogeneity. So there are some parts in the tumor, which you can see on the yellow overlaps, where they infiltrate quite nicely. There are other areas where they do not infiltrate so nicely. So that's interesting. Here is ex vivo validation of all of this. You see in the brown stain the tumors, and you see here on top on the brown stain the gamma delta T cells. And depending on the administration of the mevalonate pathway inhibitor, you can see that we can increase the number of infiltrating cells by a factor of 2 or more. That has also been translated into humans in a different context. It's an autoimmune context here. So people in Birmingham, this time it's the Birmingham in UK, were interested in liver homing of clinical grade regulatory T cells after therapeutic infusion, and they achieved this with indium tropolone. You see quite nice images. For a few days, they can follow the cells. It's brilliant. And this regulatory Tregs is something that is quite a big thing. It's being developed, co-developed at King's with a large consortium of people and led by Giovanna Lombardi. And they are really envisaged as a cell therapy for alleviating transplant rejection. And there's lots of trials that have been already run. And there is science for safety, and they are safe. And there's also efficacy demonstrated. And this induction of tolerance, which is envisaged to be achieved by these Tregs, is something that is not unique for the transplant rejection arena, but they are also envisaged nowadays as an immune therapy for autoimmune disease, for transplantation, graft versus host disease, and even more as they get engineered with CARs and become so-called CAR Tregs. And so we are working in this space. And but what we need for that arena is that we need to come somewhere closer towards tracking them for a longer period of time. And one of the aspects that is critical in that context is, of course, always going back to your textbooks and thinking a little bit about the laws of physics. So if you can track something for 1 week and you need a certain amount of radioactivity for this, and you want to extend the tracking time, you need to think along the lines of radioactive decay load. So if your threshold of your PET detector sensitivity stays, of course, constant, it means you need to significantly and exponentially increase the amount of radioactivity that is your starting radioactivity. And the question is, can your cells survive this? So we radiolabeled at different amounts these Tregs and quite high amounts. And looked into what they can take, how much can they take, how long would we be able to track them, would they remain functional? So this is just characterization on this side. So the really interesting image is what you see here. So when we administer these cells into even naive animals, you see after an hour, you see the typical image, a load in the lung. 2 days later, a load in the liver areas. Nothing in the lungs was all good signs. But when you start to look closely, particularly a few days later, start to look more closely, you see lots of bone signals. And this is not where these Tregs should be. And when you start to analyze in those animals various types of bones, like pelvis, femur joints, left and right, and so forth, you start to realize that actually over time there is increasing bone uptake. So what does this tell us? Is this something of concern? Well, yes, it is. Zirconium has an ability, if it is a free isotope, to get into bones. It gets metabolized into bones, if you like. And it's a sign of free zirconium in the system. So the question is, where does this free system zirconium come? So it could be a little bit of efflux, but it's something much worse in this case. Because of the high radioactivity amounts in those cells, they look viable after radiolabeling, but they are not able to expand, as you can see from all of those curves here. At those levels, like 100 millibecquerel, 300 or 600 millibecquerel, and these are the images from 100 millibecquerel. So in other words, when we bring this, when we synthesize this with our original goals of 2, 3, 4 minimum weeks of tracking with these longer half-life isotopes, that's not going to work ever, simply because the Tregs are too radiosensitive, and with the radioactivity that you need for the long-term tracking, you are way off the amounts that they can sustain, and they will die. And as a consequence, you see the zirconium in the bone. And you may argue that, hello, total body PET comes up, and people say you can gain an order of magnitude. Yes, you can gain an order of magnitude, but you, for like 3-4 week tracking, you are closer to 2 orders of magnitude away. So that doesn't look overly good. As a consequence, we have switched into tracking with reporter gene labeling. So what is reporter gene labeling? You basically put a reporter gene into your cells, and similar to GFP, but the difference is then that you add a contrast agent. And that basically picks up the cells you have put in with your reporter on the basis of the reporter. And you now use really short half-life isotopes so that you don't put too much dose into the cells or into the subject. And that should enable repeat imaging. How do you get the reporter in? Well, the conventional genetic engineering technologies, either viruses, episomal plasmid, transposons, gene editing, whatever you like. One of the main advantages is now you have cell viability information, and you have indefinite observation times because obviously this is passed on to the filial generations, and you keep in mind short half-life radioisotopes. So this is just, as I know that you record, so you can afterwards look at that slide as a table of human PET reporters. Today, I will largely talk about NIS, which is a non-immunogenic host reporter, and which uses radioisotopes or radio tracers that are exclusively renally excreted. So here's an example, an overview of this. Again, it's a host reporter. It's highly restricted in re-expression, meaning that there's lots of organs. You can use it for tracking things. You have renal excretion, and when we actually operate in the preclinical space, then we have a tendency to fuse it to a fluorescent protein, which is possible and which is really practical for the generation characterization of cells, and that marker is then also useful for ex vivo analysis on the tissue level in preclinical settings. How does that look like? Here's a typical example. You saw here a tumor here that metastasized, for example. Here's cancer cell tracking to the lungs. It's quite impressive how this works. You can obviously quantify, segment and quantify each and every of those lesions for the volume of where the radioactivity sits, for the amount of radioactivity that's in there. And because of that fluorescent protein, we can do histology too. So it's a very nice technology. You can read up how that works for cancer cell tracking in all of those papers here. But we use the technology for tracking Tregs. So the first question was, of course, is it possible to engineer these Tregs so such that they keep their phenotype? Well, yes, we can express it in those Tregs after viral expression for several weeks without any changes. Reporters are functional, phenotypes remain. And most importantly, also the suppressive function, the immune suppressive function of the Tregs is sustained, no matter whether we label them under conditions that are similar to in vivo labeling or not. And that is very, very positive. So we are no longer killing the cells, yeah, like we did with the zirconium-oxime. So how did we use this? If we use it for regular T-cell therapy trafficking to transplant. So this is a human skin transplant on a mouse, an immune-compromised mouse. And after engraftment, the Tregs were administered to these mice, and we were looking whether they are, at what time they are actually arriving at the transplant. You can see that after about a bit more than a week, you see quite nicely the arrival by SPECT CT here of the traceable Tregs. And we can, of course, do histology, and you see here the quantity. So basically, by microscopy, with the markers that these are indeed Tregs and human Tregs, because as you can see on the human CD3 staining in red and the FOXP3 staining as a marker for the regulatory T cells in green. You can do quite interesting experiments with this. So, for example, the trafficking of Tregs is dependent on inflammatory cues, CCL17, CCL22. If we interfere with this system and make neutrophils no longer be able to go to the transplants because we deplete them, or at least we deplete the GR1 positive population amongst them, then we also interfere with the Treg tracking towards the transplant. So, and as you see, these are studies very informed by imaging. Imaging is a readout, so it's quite a powerful tool to learn something when you, for example, were interested in conditioning therapy of your Treg to make them better or precondition the subject and things like this. Yeah. So what else are we doing with those? I mentioned briefly that obviously the field is going towards CAR Tregs. So we were involved in the generation of the UK version of the HLA-A2 CAR Tregs. You see here how HLA-A2 CAR Tregs, how much better they go to a transplant of a transgenic mouse that has a human HLA in the transplant as compared to the Tregs that are not HLA-A2 CAR Tregs and just polyclonal ones, so it's quite impressive. So we have demonstrated that you can efficiently engineer these Tregs, that they do not differ in phenotype from untransfused Tregs. You can use this imaging to detect them. The imaging per se doesn't impair the Treg phenotype or function, and we've delivered them in vivo this Treg tracking across several types of models. So one of the next steps that we are really envisaging is to actually try this in the clinic in real patients. So what else are we doing along those lines? Well, we are trying to supercharge this HLA-A2 Tregs, so basically using track concepts, co-expressing immune boosters, if you like, for the suppression, like in this case IL-10, make these boosters antigen specific in terms of release and so forth. And here is an example of how that actually benefits in an XVHD model and how you get on the orange curve better transplant survival than compared to the green one. So there's a lot of things that you can do in engineering cell therapies, and that is obviously very practical to have some form of imaging readout along those lines. Well, so I've showed you this now for Treg therapy. I think for the last couple of minutes, I show you that this is also possible for CAR Treg therapy, of course, in oncology. So without going too much into specific details, this is just more to show you what all you need to do in order to validate that your reporter doesn't do any harm. Obviously, you need to show that it's functional, that it doesn't impair the anti-tumor function, which it doesn't. It shouldn't impair proliferation, which it doesn't. It shouldn't cause any damage by imaging, which it doesn't. And you should be able to detect it. And we can actually detect about 3,000 cells per million cells volume, which is pretty good. It's a little bit lower than we can achieve with cancer cells, where we get in the higher hundreds. But it is also down to the size and the ability to express the reporter gene in those cells. So what can we do with this? For example, here we were interested in the persistence of CAR T cells in triple-negative breast cancer models. You can see after administration, and really we were wanting to decouple this from the infiltration. So we were administering the T cells into the tumors and then watched what happens in these T cells in the tumors. So these CAR T cells quite nicely distribute in this model here on the top. And then you see after 2 weeks, you see shrinkage, while there's still plenty of those CAR Tregs in the tumors. And you can see here on the right side, actually, how the curves start to differ. And when you look in a different type of tumor model with a lot more PD-L1 expression in vivo, you see that the CAR T cells do not distribute and not infiltrate within the tumor into other regions, and they die off within about 2 weeks' time. So the immune environment here heavily impacts on those CAR T cells, and imaging of those traceable CAR T cells is your readout. It's a very valuable tool again for the cell therapy development community.
Sorry, I would have to ask you to please wrap up. We're pretty much at the end of the time.
It's a very good point to wrap up, because that's my slide for showing you the differences between direct labeling versus reporter gene imaging. And you see here on top, well, very different picture than on the top in the bottom. In the bottom, the reporter genes, this is where the reporter gene traceable T cells expand in the right tumor. And on the top, you see they end up in the liver and possibly some in the bones, and you basically look at the wrong thing. So when you want to go for longer term tracking, keep in mind it's all about reporter gene imaging, less much about radionuclide imaging along the lines of direct labeling. And let's switch to the acknowledgement slides here. So these are the people who we are collaborating with. And importantly, also have a look at our funders at the bottom of that slide. And my most thanks go to my team and everybody who contributed to the works that I've shown you today, particularly those here in bold. So Alessia is now at MSK Memorial Sloan Kettering. Deal is at a cell therapy manufacturing plant in Utrecht. Candice is postdoc in Edinburgh. So they all have very successful careers, and I'm very proud of them and their work. And not only I would like to thank them, but obviously also you for staying on. And if you have any questions, like Tom said before, because of the time limits, just text me or put it into the chat here. Yeah, thank you very much.